Hey folks, Mr. Dunbar coming to you pre-recorded from my basement again. Picking up on where we left off with the DNA chapter. Hope you guys are all doing well, and if you're in Dunbar's class, I hope that we can see each other soon. If you're in the Hazernall family or the Ward family or the Summers family, they hope they can see you all soon as well. So hope you're doing all right, and we're going to bang out some more of these slides from chapter 12 and 13. On PowerPoint 1 of chapter 12, we got through the first about 19 slides, I believe. I did make a misstatement on the first slide. I think I was talking too fast. I'm going to correct some of that. And then we're going to go through today part 2, which is going to be structure of DNA, how that double helix forms, that twisted ladder that you're also used to seeing. It's right here up on the top left corner. And then also how DNA is able to copy itself so that we can pass our traits on to future generations. So before we do anything else, let me kind of correct a mistake I made earlier. I'm going to get my mug shot off the screen here. Again, I'll see you at the end of the video, though, okay? So if you look here, what I said the other day on the part 1 of the video is we were talking about what DNA actually does, what it actually is all about. And what it does is it actually gives you instructions to have certain traits, right? And traits are all about proteins, really. Proteins are everything in biology. Proteins serve 2 purposes, really. They can build things, or they can serve as enzymes, which help chemical reactions take place in your body, which also ends up giving you a certain trait. So when I misspoke the other day, I compared blue eyes with brown eyes. And I think I said blue eyes twice instead of saying brown eyes once and blue eyes the second time. So what I want to clarify is the way DNA works is, you guys all know DNA ultimately gives you your traits. We all know that, right? But what you probably don't realize is, let me choose a new cursor here. What you probably don't realize is how DNA, the organization of the molecule you have in every nucleus of your cell, and all living things have, by the way, how does that actually lead to a trait? So we talked about brown eyes, for instance. So here's how this works. The simple thing is the DNA for the brown-eyed, gene, you could call it, leads to your brown-eyed trait, and you all know that. But there's really some important steps in the middle that you probably don't realize. And here's how the big picture works. The DNA that somebody, let's say with brown eyes, has would be a certain code or certain order of those bases we call adenine, thymine, guanine, and cytosine. Don't worry about that just yet, but you did get a little tease of that in the first PowerPoint. You saw them as letters, A, T, C, and G, but they're really just chemicals. If you have a certain order of base chemicals, A, T, C, and G, in your DNA, really what that does is it passes its message off to a helper molecule called RNA, another nucleic acid, which is where the Na comes from, nucleic acid, signifying that it comes from out of the nucleus. It's in the nucleus of all cells. This is very, very tiny stuff, very much smaller than a cell. So DNA, your code for brown eyes, really passes that code to RNA, which gives instructions on how to build a protein enzyme that's going to give you a chemical reaction that gives you brown pigment, which is going to give you brown eyes. If you have the trait or the gene for blue eyes, you're going to have a different spelling or a different order of your letters in your DNA code, which is going to pass off a different message to RNA, or copy a different message to RNA. And that RNA is then going to make a different protein, because that's a different instruction, which is going to really, these proteins are enzymes, that's going to give you a different chemical reaction that leads to blue pigment, that leads to a blue-eyed trait. So if I messed that up in the first PowerPoint, or if I went too fast or spun you around a little bit, I apologize. So let's get to the bigger things here. We've been through the first, what we say, 19 slides, and this whole first part of the chapter in 12.1 is really about the history and discovery of how we found out DNA was the responsible molecule for passing on inheritance and giving us our traits and our information. We talked about Frederick Griffith, we talked about what he did with these mice, and we talked about a couple other folks, Hershey and Chase and Oswald Avery, and we talked about what they did with viruses and bacteria, and I'm not going to go back over that again. But what I did want to show you that I didn't have time to do earlier was when we talked about how viruses infect cells, we also started talking about how this is appropriate to the coronavirus that's going on right now. The way viruses work is viruses are not technically living things. They're very close to being alive, but they're technically not alive because they can't reproduce on their own. They need a host cell to do that. If they had their own reproductive machinery, they would be considered alive. But because they are so dependent on other cells, they are considered close to living, but not technically alive. So the way viruses work, and this is what Hershey and Chase were studying, but again, this is timely because of the coronavirus, is what viruses do is they really just inject instructions into other cells, and they take over that cell's machinery. So it's much like a virus you would get on your computer. It injects instructions into your computer and starts running your computer and saying, make more viruses, which is what makes viruses very, very good at what they do. But what do they actually pass into the host cell in order to control it and to take over its machinery and to give it its own instructions? Well, they really just pass genetic information, either DNA or RNA. That's what viruses do. So here's a quick little video to show you how they do that and why they're so good at what they do. Okay, remember, phages are viruses. So that's the virus right here landing on a cell. In this case, it's a bacteria cell with just one single loop chromosome of DNA. Remember, bacteria don't even have a nucleus, they're prokaryotes. So this virus literally looks like a little spaceship, a little moon lander or something from Apollo missions. And it's got protein, and it's got a loop of DNA or RNA, in other words, instructions in here. And watch what it does. This is pretty neat. It actually injects its instructions, injects its DNA right into the bacteria's DNA. And boom, what that did was that told the bacteria, make more viruses. So these are all brand new baby viruses. And those viruses would then go on to infect other cells and repeat the whole process. That's what makes the coronavirus so dangerous, and actually makes really all viruses potentially dangerous. But we can also use that as a way to insert good genes to replace bad genes for disease. We also talked about the way they've used that virus concept to insert genes from a lightning bug into a mushroom. What they did here was, we talked about this earlier, they took a gene out of a lightning bug, they put it into a virus, they let the virus infect the mushroom, the virus makes copies of itself inside the mushroom, or really tells the mushroom cells to make copies of the virus, because that's what viruses do, they take over. Much like a pilot hijacking a plane, or again, like a viral software that you might have in your computer. And it says make more viruses, and boom, that virus actually was inserting the gene that the scientists stuck in there from the lightning bug. So it was really a clever way for scientists to deliver a lightning bug gene or instruction into a mushroom and make it glow like a lightning bug. So imagine what we could do to help heal diseases. If, for instance, we had, let's say, somebody with diabetes, and instead of wanting to treat them the rest of their life, could we just fix the gene that is wrong with diabetes? It's a gene that doesn't allow you to produce insulin many times. And could we take a good gene and stick it into a virus, and stick that virus into a person, and have the virus deliver the good gene so that the person now gets the instructions on how to make insulin, and they're no longer diabetic, and that would be a cure. There's a lot of work being done with this, these bio-targeted therapies, they call them. Okay, moving on. So we got all past this stuff. Hershey and Chase figured out that it was DNA that was the responsible chemical for passing on genetic information, and then also leading to how we build our traits, how we get our chemical reactions, and how we build our proteins. So this was where we ended up last time, I believe, slide 19. And you guys got a little tease of the double helix here. Remember, the double helix really just means a twisted ladder. It's like two spiral staircases wrapped around one another. And it's a beautiful molecule. It's made of four very simple chemicals in the middle, and then a couple more chemicals on the ends. And here's what it's made of. You see the steps on the ladder here. These are the A, T, C, and G. Again, think of these like letters in a sentence or something. Remember, DNA is like a code book. It's really like a whole library of code book or instruction book. Thymine, adenine, guanine, cytosine. So these are our four choices, and this is all living things, from a bacteria to a tree, to your dog, to a spider, to you. We all have the same 4 base letters in our genetic alphabet. What makes us different is the order that we put these in. We can make a lot of different instructions by putting these letters in different orders. So whatever is down one side, we said this the other day, will determine what is along the other side, because A always pairs with T. It looks like if you look in this diagram here, you'll always see a red bonded to a green. Every time there's a red, there's a green next to it. Every time there's a green, there's a red next to it. Adenine always bonds with thymine. In my class, we say apples grow on trees, or trees produce apples. So T and A go together, or A and T go together. And then if you look, you always see a purple with a yellow every time, and that's guanine and cytosine, 2 different chemicals. Again, think of them as letters in a sentence or in an instruction manual, a sentence of an instruction manual. And G and C always go together. Guanine always goes with cytosine. So C goes with G, G goes with C, however you want to say it. This in my class, we said cows eat grass. So G and C. Cows eat grass, apples grow on trees. A and T, G and C. The fact that these 2 chemicals only bond in certain ways, or really these 4 chemicals, A always with T and C always with G, means that if you have the left side of a DNA molecule, you can very easily replicate it by putting on a new right side. So if I know that this is A, I know this has got to be T. If I know that this is over here G, I know that that's got to be C, and so on. This base pairing we talked a little bit about the other day is what allows DNA to copy itself. Well, who cares if DNA can copy itself? Well, that's how we pass on our traits. If DNA could not replicate or copy, it wouldn't be able to pass on traits to new generations, and everything would go extinct. So it's pretty simple, but it's pretty amazing as well. So structure of DNA is a double helix. How do we know that? By the way, here's just kind of a reinterpretation of that same model. The famous double helix is 2 intertwined sugar and phosphate chains. The sugar and phosphates are the sides of the ladder. So where you see the blue and light blue here, these are the sugar and the phosphorus or phosphates, okay? Those just kind of hold the ladder together. The blue and the light blue is not where your genetic code is, because that never changes. The genetic code, what makes us all who we are, is whatever's in the middle here. Like the steps of the ladder give us our uniqueness or diversity. Okay, so the famous double helix is 2 inverted sugars, sugar and phosphate chains linked by 4 kinds of chemical bases. Again, this is repetitive. A and C, G and T. A always goes with T, and G always bonds with C, which make up the genetic code. Again, 4 little letters. If you think about the English alphabet, we have 26 letters to choose from to write our thoughts and in our books and our paragraphs and our sentences. DNA only has 4 letters to choose from. So how do we get so much variety? Well, these chains are very, 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 very long. Very, very long in all individuals. Human beings, for instance, if you looked at all of our DNA on all of our chromosomes, because that's really what chromosomes are, they're just chunks of DNA, we have about 3 billion base pairs. So 3 billion steps on our genetic ladder. And remember, that all fits into the nucleus of every single one of your trillion cells. So this DNA stuff is tiny, 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 but contains a ton of information. A ton of information. So structure of DNA. This is kind of a conceptual model, right? But let's take a look at if you could see the actual atoms involved here. So let's break it down a little bit further here. DNA structure. DNA is a large molecule made up of units called nucleotides. You guys have learned in biology that structures always contain like building blocks. The building block of DNA is 1 nucleotide, and I'll show you what that is. Each nucleotide contains 3 parts, just like you kind of saw up here in this ladder. I apologize. Every single nucleotide contains one of these little white balls here, one of these little gray balls, and one of the 4 colors. So one of the 4 base pairs, A, C, T, or G. These bigger white sugars, you can see from up here from the key, is called a ribose sugar or a deoxyribose sugar. And this little guy is called the phosphate. And again, those never change. The only thing that changes is the base pairs. And 1 nucleotide, 1 building block, contains 3 pieces. Always one of these sugars here, the big circle, always one of these phosphates, and then also one of 4 bases attached to it, whether it's the A, the C, the T, or the G. So that whole unit, 1, 2, 3 would be called a nucleotide. Here would be another nucleotide. Pick one. 1, 2, and then the yellow base, thymine. That would be a thymine nucleotide. Go over to here. A phosphate, a sugar, and a guanine. That would be a guanine nucleotide, that little triplet right there. So nucleotides contain 3 parts: a 5-carbon sugar called deoxyribose, a phosphate, so that was the big circle, that's the little circle, and then one of those 4 bases, A, T, C, or G. And here's what it looks like if you enlarged it. You would see this phosphate, the phosphate groups here, and then the sugar over here, and then one of the 4 bases over here. Don't worry so much about that yet. And of course, we know that A always goes with T and C always goes with G because DNA is double-stranded. A little bit more detail here. Not sure how important this one is, but you may see this down the road. If you look, you already know that A and T go together and C and G always go together. But if you look closely at these molecule structures, this is kind of how the atoms in these molecules are bonded together and the shapes that they take up here. Notice that adenine and guanine have 2 ring type structures to them, these like pentagon, hexagon rings, whereas thymine and cytosine only have 1 each. Well, a big one, a small one with only 1 hexagon or pentagon, will always bond with another base pair that has 2 rings to it. This is why the DNA chain is always equal width all the way across. In other words, all the steps are the same width or the same size, because a large one always bonds with a small one. So in other words, if you put these together, adenine having 2 rings, which is a large base, with thymine having 1 ring, which is a smaller base, the total length of those would be 3 rings long, if you like, okay? If you take a guanine and a cytosine, again, guanine is one of the larger ones, with the 2 rings there. Cytosine is one of the smaller ones. It has 1 ring. That would also be 3 rings wide. So if you look on the genetic ladder, the double helix, notice, even though these are different chemicals, the yellow, the blue, the green, and the red, the adenine, thymine, guanine, and cytosine, the width of this ladder is always exactly the same because it's always a 2 ring across from a 1 ring. All right, and the technical names for this stuff is the ones that have adenine and guanine, the ones that have only 1 ring to them, 2 rings to them, are known as purines. The other 2 bases are cytosine and thymine, and they are known as pyrimidines. So cytosine and thymine have 1 ring, they're known as pyrimidines. And adenine and guanine have 2 rings, and they are known as purines. Okay? And a purine always bonds with a pyrimidine. So purines have 2 carbon rings in their structures, and the pyrimidines will have only 1. So here are my purines, here are my pyrimidines, and a purine always bonds with a pyrimidine, which is why cytosine always bonds with guanine, and thymine always bonds with adenine. Again, don't worry so much about that, but it makes sense that the smaller ones always bond with the bigger ones, which is why the nucleic ladder, the twisted ladder, is always the same width. And here's a blow up of that same concept right here. Don't worry about these numbers and stuff yet. We'll get into this. This is called 5', 3'. Don't worry yet. But this is the side of the ladder right here. And you see these phosphates, the P, and you see these pentagon-shaped sugars. Those are those deoxyribose sugars. And again, this is the side or frame of the ladder. There's no genetic code there. That's just a framework to hold the genetic code. Okay, so the outsides don't play a role in your genetic traits. The inside, the pairs that are making up the ladder steps are what actually give us our code. And notice, you always have a double ring bonded with a single ring. A double ring bonded with a single ring. So here's a double ringed guanine bonded to a single ring cytosine. Here's a double ringed adenine bonded to a single ring thymine, which gives the ladder the same width all the way across. You wouldn't want a ladder that's getting thinner and then fatter and thinner and then fatter. So these steps are always the same distance across. However, when I twist this, it may look to you, because remember the double helix is twisted, it may start to appear to you that the ladder gets thinner and fatter, but it really doesn't. That's just kind of an illusion. Okay, structure of DNA. Again, this is going to be pounded into us. The nucleotides on one side can join together in any sequence. This is why we have variety in the world, in every organism, from top to bottom. So it doesn't matter what the sequence is going up and down, but whatever the sequence is, let's say on the left side going up and down, that will lock in or determine the sequence on the right side, because you know that if there's an A on the left, there's going to be a T across from it. If there's a C on the left, there's going to be a G across from it, and so on. And again, this base pairing rule, C with G and A with T, is what allows DNA to copy itself. And that's what we're going to get into in a little bit. Okay. Don't worry again about these 5' and 3' direction things. Think of this as DNA being a southbound lane on one side of the ladder and a northbound lane on the other side of the ladder. So kind of like a two-way street. Think of it that way. Okay. All right. Big picture here. The sequence on one side of the ladder, one side of the helix, dictates the sequence on the other side. Dictates means determines, kind of locks it in, right? If you got peanut butter over here, you're automatically going to have jelly over here. If you have ham over here, you're automatically going to have cheese over here. So if I know the left side, I can easily tell you what the right side is. And DNA uses that little trick to copy itself. Okay. Here's a great little analogy to the double helix. This is a double spiral staircase, and I believe this is at a theme park. This might be Kings Island or something. So the problem that scientists had, though, remember, they're still trying to each kind of contribute to figuring out this molecule we call DNA, and this started back in really even in the 1800s. So the problem that scientists had is, based on what they knew, they didn't really know why DNA was much different than the other compounds that they knew that all living things had. You guys learned these in chapter 2. Everything also has carbohydrates, lipids, and proteins. DNA is one of the nucleic acids. So there's 4 things that all living organisms contain, 4 big macromolecules, and they are carbohydrates, lipids, proteins, and nucleic acids. You guys have heard of those the whole year. So scientists wanted to know, well, what made DNA different than these other structures or these other molecules that all living things have? In other words, how come genes aren't made of carbohydrates? How come I can't pass my traits on using carbohydrates or using lipids or using proteins? What is it about DNA that allows it to store and pass on information? So what makes it special? And here's our old friend, Edwin Chargaff. And here's what he discovered. He started doing some research on DNA. He discovered that the amount of guanine was always equal to the amount of cytosine in any organism's DNA, no matter what, a bacteria, a tree, a person, a spider, a monkey, you name it. So he said, all right, why is the amount of guanine always equal to the amount of cytosine? And he also discovered that the amount of adenine was always equal to the amount of thymine. And now we know today it's because guanine always bonds with cytosine, cows eat grass, and adenine always bonds with thymine. Apples grow on trees. But he was the one that let us start to figure this out. Picture it this way. If you went to somebody's refrigerator and you didn't know them, and you saw, every time they bought 150 jars of peanut butter, they bought 150 jars of jelly. And every time they bought 85 slices of cheese, they bought 85 slices of ham, you would probably start to infer or figure out that they must always put peanut butter with jelly, and they must always put ham with cheese. Okay, I hope I told you they bought 85 pieces of ham and 85 pieces of cheese, as well as 150 jars of peanut butter and 150 jars of jelly. Okay, so here we're Chargaff. Again, his name is Edwin Chargaff. Here is what he found. Basically, here's a bar chart, or bar graph. Adenine and thymine were pretty much equal in amounts. And again, this is for any living organism, not just people. He also found that cytosine and guanine were basically equal amounts. Okay, so the amount of peanut butter and jelly that you're using is equal, and the amount of ham and the amount of cheese is equal. So ham must go with cheese, and peanut butter must go with jelly. That's how we figured out that A goes with T and C always goes with G. You'll notice here on this graph, they're not quite equal. Look very closely. The amount of adenine looks to be slightly more than the amount of thymine. And one reason for that is because sometimes there are misspellings in DNA. And there are mistakes. And so these two could be slightly different. These two could also be slightly different. Once in a while, you might make a ham and peanut butter sandwich by accident, and you might make a jelly and cheese by accident. It kind of screws up your numbers. All in all, though, these numbers total will add up to 100% of the base pairs in an organism's DNA. Remember, base pairs are the two pairs on a step, or what we call a rung of the ladder, a base pair. And humans, again, have about 3 billion base pairs that give us all our information. So after Chargaff, Rosalind Franklin, and this lady was a very special lady, and she kind of got the shaft a little bit in science. She was living at a time when female scientists weren't very well respected yet in their fields, and she also really was instrumental in discovering the shape of DNA, but she didn't get credit for it. She kind of got railroaded by a couple of other people, a couple guys named Watson and Crick and another guy named James Wilkins. I'm going to show you a very quick video about her in a second. So early 1950s, Rosalind Franklin was studying DNA, and what she did was she took x-rays and she bounced them off of DNA. She shot them at DNA, and she kind of saw what the pattern was of the X-ray, what we call diffraction. So it's like almost like an echo or a bouncing off of X-rays. This didn't really answer the question about what DNA was made of or the exact structure, but it did give us a clue as to the shape of DNA. And here is her very famous photo. This is in a museum in England now, I think. This is called Photo 51. This is Rosalind Franklin's X-ray picture from the 50s of DNA. And you can kind of see, it kind of gives you the shape of a helix if you looked at it from the side, okay? Or actually down the length of it. This Xing pattern is really two twisted ladders, or a twisted ladder with two spiral helixes, a double helix. The biggest clue revealed was that DNA strands twisted around each other like a coil. This shape is called a helix. DNA is a double helix, all right? So DNA is very, very efficient at putting a lot of information into a small space. Part of that is because it's a double helix. It's a very good way to put a lot of stuff in a small spot, which nature does very, very well, okay? Now, before we go on to Watson and Crick, quick background here. Watson, I think this one's Watson, I can't remember. Watson's still alive. Crick passed away a few years ago, but Watson's about 98 years old or something crazy, and he's still working. And at the same time, Rosalind Franklin was doing her DNA X-rays, just getting pictures of DNA. These guys were trying to work out what the exact structure of DNA was. Although these guys kind of ripped her off, and I'll tell you that story in a second. And Watson and Crick, they were physicists. Francis Crick was a British physicist, and James Watson was an American biologist. And they were trying to understand the structure of DNA. And this is them back in the 1950s in their lab, and they're actually building a model of metal pieces, trying to fit all the base pairs in and seeing if it would work, and seeing if they could kind of recreate the structure of DNA based on what they did know, the limited stuff they did know. So they built a three-dimensional model of DNA, but no matter what they did, up until the point that they got Rosalind Franklin's photograph or her X-rays, they couldn't make it work. They really didn't understand it. It was her photograph that really unlocked the whole situation. So kind of a good science mystery, all right? So Crick, that was this guy on the right here, I believe, got a hold of the work that Franklin was doing. In other words, he didn't steal her photograph, but he borrowed it without her permission, put it that way. And within weeks of seeing it, he figured out the structure of DNA. And of course, he figured out that it was a double helix. Let me show you this story really quick.
The discovery of the structure of DNA was one of the most important scientific achievements in the last century, in human history, in fact. The now famous double helix is almost synonymous with Watson and Crick, two of the scientists who won the Nobel Prize for figuring it out. But there's another name you may know too, Rosalind Franklin. You may have heard that her data supported Watson and Crick's brilliant idea, or that she was a plain-dressing, belligerent scientist. which is how Watson actually described her in The Double Helix. But thanks to Franklin's biographers, who investigated her life and interviewed many people close to her, we now know that that account is far from true, and her scientific contributions have been vastly underplayed. Let's hear the real story. Rosalind Elsie Franklin was born in London in 1920. She wanted to be a scientist ever since she was a teenager, which wasn't a common or easy career path for girls at that time. But she excelled at science anyway. She won a scholarship to Cambridge to study chemistry, where she earned her PhD, and she later conducted research on the structure of coal that led to better gas masks for the British during World War II. In 1951, she joined King's College to use X-ray techniques to study the structure of DNA, then one of the hottest topics in science. Franklin upgraded the X-ray lab and got to work, shining high-energy X-rays on tiny, wet crystals of DNA. But the academic culture at the time wasn't very friendly to women, and Franklin was isolated from her colleagues. She clashed with Maurice Wilkins, a lab mate who assumed Franklin had been hired as his assistant. But Franklin kept working, and in 1952, she obtained Photo 51, the most famous X-ray image of DNA. Just getting the image took 100 hours. The calculations necessary to analyze it would take a year. Meanwhile, the American biologist James Watson and the British physicist Francis Crick were also working on finding DNA's structure. Without Franklin's knowledge, Wilkins took Photo 51 and showed it to Watson and Crick. Instead of calculating the exact position of every atom, they did a quick analysis of Franklin's data and used that to build a few potential structures. Eventually, they arrived at the right one. DNA is made of two helicoidal strands, one opposite the other, with bases at the center like rungs of a ladder. Watson and Crick published their model in April 1953. Meanwhile, Franklin had finished her calculations, come to the same conclusion, and submitted her own manuscript. The journal published the manuscripts together, but put Franklin's last, making it look like her experiments just confirmed Watson and Crick's breakthrough, instead of inspiring it. But Franklin had already stopped working on DNA and died of cancer in 1958, never knowing that Watson and Crick had seen her photographs. Watson, Crick, and Wilkins won the Nobel Prize in 1962 for their work on DNA. It's often said that Franklin would have been recognized by a Nobel Prize if only they could be awarded posthumously. And in fact, it's possible she could have won twice. Her work on the structure of viruses led to a Nobel for a colleague in 1982. It's time to tell the story of a brave woman who fought sexism in science and whose work revolutionized medicine, biology, and agriculture. It's time to honor Rosalind Elsie Franklin, the unsung mother of the Double Helix.
Kind of a sad story. She kind of got the shaft on that one, and she was not able to get the Nobel Prize because they will not award the Nobel Prize after somebody has died. And the Nobel Prize also comes with a pretty hefty cash prize as well. I think it's $150,000 or something crazy. So she kind of missed out on that, unfortunately. So, so one more thing. She died of ovarian cancer, which they believe was a result of her working with so much X-ray radiation. So that's also kind of sad. So at least we got a lot of information from her, though, and we really have her to thank today for this. So Watson and Crick, they took her information kind of without her knowledge, but at least they did something kind of good with the information, at least. So, excuse me. Watson and Crick's model of DNA was a double helix, in which two strands were wound around each other, again, like two spiral staircases intertwined. Their double helix design explained many of the things found in Franklin's experiment. They were not able, though, to explain what held the two strands together. What held them together? Today, we know it's the base pairing and the bonds that form between the base pairs. The bonds that actually form between these base pairs are not very strong, which allows DNA to come apart, and that's important for when it needs to copy itself. These bonds that hold DNA together are called hydrogen bonds. You may have learned about these in your biology class. Hydrogen bonds a lot of times will also form between water molecules, which is what gives water kind of that rubber sheet effect, why bugs can walk on water and why water kind of behaves the way it does, because water molecules kind of stick to each other or are attracted to each other by something called hydrogen bonds. These are weak bonds, but there's a lot of them, and it holds the A to the T, and it holds the C to the G. And it also allows for temporary bonds, which can be broken and reformed very quickly. This is important, again, because when DNA copies, the strands have to come apart, and I'll show you that down here. Okay, so back to this base pairing business. Again, the base pairs are held together by what we call hydrogen bonds. Once they saw this principle called base pairing from Chargaff, again, this is Watson and Crick we're talking about. Once they saw Chargaff's rules, and then they saw Rosalind Franklin's photograph, or her X-ray photograph, they started putting it all together. All right. So now there was a reason why A always went with T, always C always was bonded to G. In other words, for every A, there had to be the exact same number of T's. Apples grow on trees, and the same is true for C and G, cows eat grass. For the amount of peanut butter, there's always the same amount of jelly. For the amount of slices of ham, there's always the same amount of slices of cheese. And the reason for that is C will form a hydrogen bond with G, and A will form a hydrogen bond with T. And there's Chargaff's big picture rules there. Make sure you know this. This is super, super critical, and one of the reasons that we have been able to work with DNA since discovering this and find out many more secrets. The fact that A always bonds with T and C always bonds with G is the reason that DNA can replicate or copy itself. Again, a big picture thing. Make sure you know this. Make sure you know this. Okay. All right. Quick comparison of eukaryotic and prokaryotic DNA. I mentioned a little bit of this in the beginning. No matter if you're a eukaryote, which is generally more advanced things like plants and animals and fungus, or if you're a prokaryote, things like bacteria. These are very simple. Eukaryotes are more complex, like humans and stuff, right? Remember, eukaryotes have a nucleus with DNA in it. Prokaryotes have DNA, but they don't have it protected in a nucleus. In my class, we say if the DNA is like the mayor, the nucleus is like the city hall that the mayor stays in. So prokaryotes, they have a mayor. He just doesn't have a city hall building to go to work every day. He floats around in the city. Eukaryotes protect their mayor within a city hall, but they both have a mayor. They both have DNA. Okay. All right. So eukaryotic cells are obviously more complex, right? They're the things you can typically see with your naked eye. So again, like funguses, mushrooms, molds, any kind of plant, any kind of animal, typically. Things you can't see are typically prokaryotes, like bacteria and other small little organisms. So eukaryotic cells are more complex. They're much more advanced, and many times have thousands of times more DNA than their prokaryotic counterparts. That makes sense that you have more DNA than a bacteria, I guess, right? Prokaryotic, in other words, bacteria mostly, cells don't have a nucleus, and they are much simpler. They still have DNA, but it's a little less complicated. For example, bacteria, instead of having those X-shaped chromosomes like we saw in the beginning of this unit and you saw in the genetics unit, they just have a single loop of DNA. It's just a circular loop of DNA. And bacteria can do weird things with their DNA. They can exchange it with other bacteria and all kinds of weird stuff. It's called conjugation, and we saw something called transformation in the beginning of this video. So bacteria can swap DNA directly. Humans and other eukaryotes can't really do that. We can combine DNA. For instance, when we have babies, half the DNA comes from the mom and half the DNA comes from the dad. We can combine DNA that way. But bacteria are asexual. They don't have sex to reproduce. They're basically clones of one another, carbon copies of one another, but they can from time to time actually switch pieces with other bacteria. Kind of a strange concept. So either way, DNA in both bacteria and in eukaryotes, so in prokaryotes and eukaryotes, still have the same four letters or the same four base pairs in their genetic code, in their letters in their genetic alphabet, A, T, C, and G. And not only that, they follow the same rules. A is always going to go T, C is always going to go with G. I hope you're picking up on that concept by now. I've said it probably 1,000 times. Sorry. Okay. E. coli, probably something you've heard of. It's a bacteria. It actually is a shortened name. It's a scientific name. Escherichia coli, I believe it is, and this stands for coliform. It's just a fancy name for a type of bacteria that typically lives in people's intestines. You've probably heard of this in food poisoning. If people's bacteria from their intestines get onto your food, it can make you very sick. If you looked at that bacteria and you looked at how many steps on the ladder it has. So if you unwound all its DNA and counted the actual steps that it has, really you'd be counting base pairs. That's what the steps are. It has about 4,600,000 base pairs, right? Remember, humans can have thousands of times that, right? The length of DNA molecules in E. coli is about 1.6 millimeters. That's actually kind of big. It's about the thickness of 2 dimes. Typical E. coli is 1.6 microns. That is 1.6 millionths of an inch. So you could see the DNA is actually a lot bigger than the bacteria that it's in. Well, how is this possible? How can you have something that's millimeters in size fit into something that is microns in size? In other words, this is a very large unit, believe it or not. This is a lot smaller unit. I can see a millimeter with my naked eye. I can't see a micron with my naked eye. So how are we able to get all this DNA into such a small spot? I'll come back to that in a sec. A human cell has 1,000 times as many base pairs as a bacteria. It's been estimated that a human cell has 30 billion total base pairs. Even the smallest chromosome has up to 30 million base pairs. And remember, humans have 46 chromosomes. Some of them have 150 million base pairs each on them. So total base pairs, total steps on the genetic ladder of a human is about 30 billion, okay? This DNA, even in, this is in all cells. Even in one human cell, if you unwound the DNA from inside the nucleus of any cell in your body, pick a skin cell or something and unwind the DNA from inside the nucleus of that skin cell, and it's almost 2 meters long. It's about 6 feet long. So how can I fit a 6-foot chain of DNA, a double helix, that's over almost 2 meters, 6 feet? How can I fit that into a microscopic nucleus of one of my cells, let's say in my skin or in my brain or in my liver or something? Well, it's actually very, very possible. And the reason it's possible is because you wind it up. Just like you could fit, 300 feet of string into your front pocket if you wound the string up. That's how DNA works. It winds up. Remember, Mother Nature does an excellent job of taking large things and putting them in small spaces. All right. This is kind of complicated, but you guys can do this. This is a kind of a diagram getting progressively more detailed as you go up. So we're going to start with the large scale stuff down here, chromosome, and get all the way down to the helix. So if you took a chromosome, remember humans have 46 of these. Fruit flies have, I think, 8 of these. Chimpanzees have 48 of these. Dogs have, I think, 78 chromosomes, right? These are very large collections of DNA. If you think of DNA like a string of letters in sentences, think of a chromosome as an entire chapter. So many pages, many paragraphs, many sentences, okay? Humans have about 46 chapters. But remember, this is very tightly coiled up. You got to fit all this information inside the nucleus, which is microscopic. So DNA really folds itself, twists itself, spirals itself, and folds back on itself again to make itself smaller and smaller and smaller and smaller and smaller in order to coil up into these chromosomes. So as I start to unwind a chromosome, I would start to see these condensed sections. We call this chromatin, and keep unwinding that. You start to see a little bit more detail in here. If I zoomed in on this, I would actually start to see DNA is wrapped around these little structures called histone proteins, which we call a nucleosome. Don't worry about all that crazy terminology. And then focus in on one of those little histone proteins. They're really like beads on a string. These histone proteins allow DNA to have something to wind itself around. If you have a string and you want to wind it up, it helps to have something to wind it up upon. If you want to roll your toilet paper roll back up, it helps to have the cardboard tube in the middle to roll it around, right? So these histone proteins are similar to either beads on a string or the cardboard tube inside a roll of toilet paper. But then if I actually look in more and more detail right here, then I can start to see that the whole time all this was a bunch of double helix DNA. So again, big picture, the entire structure all wound together. unwind it and untwist it and uncoil it and unfold it, and you get down to the DNA double helix level. So a chromosome is nothing more than a bunch of tightly coiled and wound up DNA. Okay. Letters, whole sentence maybe, right? Paragraphs, pages, entire chapter. All right. Add all your 46 chromosomes together and it gives you your entire instruction book. So I hope you're getting a feel for how this stuff's organized. Okay. All right. We talked about this already. These proteins is just a way for DNA to wind itself up. If you look close at it, not that important, but respect the way that nature has a very, very efficient way of getting big stuff into a small space. Okay. All right. New concept. Take a breath, take a break, and pause the video if you need to. Okay, guys, we're almost done. A few more slides, and actually, I think 3 slides to go, and I have 2 couple-minute videos for you, and we'll be done. Let me back up for a second before we start this process of replication, because this is kind of a brand new section now. This is 12.3 in your chapter, in your DNA chapter in your textbook. But I want to go back and give you, again, big picture stuff so you don't get too lost in the shuffle here. One second. What are genes and what are DNA and genes?
Building all living things, whether a fish, a bacterium, a tree, an insect, or a person, are found in a molecule called DNA. DNA is made up of 4 tiny building blocks. Each building block has a backbone and a chemical base that is represented by the letter A, C, G, or T. DNA molecules have 2 strands that are joined together through complementary base pairing. A pairs with T, and C pairs with G. These building blocks function together in larger units called genes. Each gene is a set of instructions for building a specific protein. A complete set of genes is called a genome. A genome is a set of instructions for building an entire organism. Every person's genome has the same genes arranged in the same order. But small differences in the sequence of the bases in our genes make each person unique.
Okay, that's important to remember. Small differences are what make us all unique. Kind of interesting. Not sure what's going on here. All right, back to our PowerPoint. One last thing here, one last video. If you want to geek out on this and nerd out on this, you can actually go to this website here. The link is built into your PowerPoint that you guys all have on your Google Classroom. And you can actually start building a DNA molecule because you know the principles of base pairing. It's kind of like a little video game. You know, it's not Minecraft, but it'll do. It's looking straight down the helix right now. So this is looking down the long end of the helix. So if you were viewing it from the top, looking down, okay? This here, of course, is a side view. But if you're looking down, your eyeballs up here, looking down the ladder, this is what you would see. Okay, that's that double helix. And you can build this and see how long it takes you. If you had a T on the right side here, of course, that would be bonded with, you guessed it, an A. Apples grow on trees, right? So A is always going to go with T. Well, here's another T, which would mean another A. Here comes a C. Which letter do I need? I need a G. Cows and grass, right? Apples, trees, cows, grass. So as you can see, this is super simple. If you know the left side, you know the right side. If you know the right side, you know the left side. This is why Mother Nature has been able to copy her DNA through living organisms for 4.5 billion years on planet Earth. It's super simple. And if it ain't broke, don't fix it. It works very, very well. That's why it has worked for 4 billion years. To pass genes on, to pass traits on. And sometimes this gets messed up and this gets spelled wrong, and we call those mutations. Sometimes I might get a tree bonding with a grass, or a tree bonding with a cow, right? Again, that's like peanut butter and cheese or ham and jelly or something. But those misspellings sometimes lead to new traits. And believe it or not, these are called mutations, but they can actually be good things as well. This is also these genetic mistakes, they add up over time and they can lead to brand new species. Remember, planet Earth started with one species 4 billion years ago. Now we have tens of millions of species, and they all came really from that first species. They all evolved from that first species over billions of years from changes, misspellings, mistakes in the genetic code that have added up over millions of years, and now we get the diversity we see today. So keep that in mind. So you can geek out on this game if you want all day long. I think once you've done 10 of them, you could do a million of them if you wanted to, right? So I think you get the idea of this. You need an A right there, and so on and so on. It shows how you're progressing down the side of the chain here, right? I'm building this missing right side or something. You can see how long this could take, but your body, all cells can do this in microseconds, very, 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 very quickly. Remember, you need to copy your chromosomes, your DNA, whenever you make a new cell, and you're making hundreds of millions of cells a day, which means you have to copy hundreds of millions of cells DNA every day just to maintain homeostasis, just to stay alive, right? To replace old cells, to grow, to heal, and so on. Okay, so this DNA replication business, it's pretty simple because you know the A to C, T to G rules, right? And we are almost done, guys. At the beginning of meiosis, so we're in this replication business now, right? And what is replication? It means copying, right? You guys just did a chapter on mitosis, when cells divide, and a special kind of cell division called meiosis, when we make cells that are only 50% the DNA of the parent. These are sperm and egg cells. So we make 50% DNA cells with a process called meiosis. This is how we make sperm and egg, and we make regular old body cells like skin and liver and lungs and so on with something called mitosis. And these cells that we make with mitosis, of course, have 100% of the DNA. These are really just asexually reproduced cells that you just double and clone to make new cells. So again, these would be like skin cells or brain cells or liver cells. But when we go to make sex cells, sperm and egg, we don't want 100% of our DNA in there. We only want 50%. So we use a special kind of cell division that reduces the amount of DNA in sperm and egg to 50%. And the reason for that is if you have an egg with 50% from a mother and a sperm with 50% from a father, the baby would be back up to 100% DNA, and then that baby would start growing by mitosis. However, either way, you need to replicate DNA in either of these processes, either of these types of cell division, you have to copy the DNA because one cell needs to keep a copy for itself, and it also needs to pass a copy on to the next cell, right? So you guys saw the details of that in chapters 10 and 11. Now we're going to talk about how that actually happened. You saw in chapters 10 and 11 chromosomes copied or chromosomes replicated. Well, how does that actually occur? Remember, chromosomes are nothing more than coiled up and wound up DNA, right? So during replication, what happens first? Well, the double helix unzips. We talked earlier about those hydrogen bonds, those weak bonds holding this ladder together. These are easily unzipped. So now I'll have a left side and a right side. Instead of a double helix, I'll have two separated single helixes or helices, right? Instead of calling it left side and right side or northbound lane and southbound lane, they actually, scientists call this the 5 prime, 3 prime, and this the 3 prime, 5 prime. Don't worry too much about that. Of course, this is interesting. In order to unzip this ladder here, you need a special enzyme called helicase. Helicase, you can tell it's an enzyme because it ends in ASE, okay? All enzymes or most enzymes end in the word ASE. Remember, enzymes control chemical reactions. So this enzyme called helicase is responsible for unzipping the DNA so that it can copy itself. Ironically, if you remember from the first slide, it's DNA that creates or gives a code for enzymes like helicase, right? There's a second enzyme called DNA polymerase, which actually allows the left side to copy itself and the right side to copy itself. Again, this is another enzyme because it ends in ASE. That's how you know that. But back to the ironic part of this, helicase and polymerase, DNA polymerase, are both enzymes. And they're needed in order for DNA to replicate and copy itself and create new DNA. So it kind of begs the question, how could the first DNA molecule have been constructed and then copied without helicase or polymerase? And don't get too hung up on these words. What I'm saying is it takes DNA to make helicase and polymerase, but it takes polymerase and helicase to make new DNA. So what came first, the chicken or the egg? This type of question will keep you up all night. It takes DNA code to make these two enzymes, but it takes these two enzymes to make more DNA code. So we may never know the answer to that, although the answer may lie in a chemical called RNA. We're not sure about that yet, but kind of an interesting concept. What came first, the chicken or the egg? They both need each other. Okay, so for replication, we're going to split these two halves apart. And remember, if we know the old left side, we can quickly build a new right side. If we know the old right side, we can quickly build a new left side because we know that wherever there's an A, across from it's going to be a T. Wherever there's a G, across from that is going to be a C. So this is very easy to do for our bodies, and they do it very quickly. All living things copy their DNA very quickly. All right, so DNA replication. Because of the base pairing principle, A to T, C to G, these individual strands can be replicated. They can be copied. In other words, if a single strand, so let's pretend we unzipped a double-stranded DNA molecule. If the one side of the strand, let's call it the left side, had a combination of, going from up to down was TAC, GTT, then the complementary strand that would be built next to that to stick to that on the right side would be ATGCAA. Now, how do I know that? Well, again, if I had TACGTT on the left side, going from, let's say, up to down, I would have ATGCAA on the right side going from up to down, because wherever there was a T, across from it would be an A. Wherever this A was, across from that would form a T. Remember, these are adenine, thymine, guanine, and cytosine, the letters or the genetic codes, okay? If there's a C here, there's going to be a G across from it in the third spot there. If there's a G in the fourth spot, there's going to be a C across from it, and so on and so forth. We would say these two strands are complementary. All right, the original right side of the DNA, this is very important information right here, guys, very important. After that original molecule split apart, after this original molecule, this double-ended molecule, double-stranded molecule split apart, the old left side builds a new right side. The old right side builds a new left side. Keep that in mind. We're going to copy our DNA based on what we call a template process. So the original right side, the old right side of the DNA helix makes a new left side. And the original or old left makes a new right. So by the end of the process, you have two double helixes, and DNA has copied itself and copied the code. And you would then be able to pass that code on to the next cell, or to your sperm or egg cells or whatever, or to your children through sperm and egg. And you would also be able to keep your original copy for yourself because you need your own instruction manual as well. So pretty interesting stuff, pretty simple when you think about it, but really, really works well. When replication is done, when copying is done, there are two double helixes or helices. Each double helix contains one strand from the old DNA and one that is brand new. Please, please, please make sure you know this. Each replicated double helix is half original, old, and half new. We call this semi-conservative replication because it's not totally a carbon copy, because each new DNA molecule is really half old and half new. So we refer to that as semi-conservative copying or semi-conservative replication, okay? And the parent DNA molecule consists of two polynucleotide chains. Poly just means many, many bases, held together by bonds. By bonds between the nitrogen bases, between the A's and T's and C's and G's. You can see replication occurring right here. This was the original double-stranded DNA molecule. This was the original, the blue. It unzips right here. There's a fork in the road right there, okay? This is now the old left side. This blue over here is the old right side. You'll notice the old left side, a green arrow here starts forming. You see this is going to be a new right side. On the old right side, the blue part here, you see the green arrow representing a new left side. So pretty soon we're going to go from having one double helix that was all original to having two double helices that are each half original and half new. And pretty soon, when this process finishes off, remember this is a human being, it's got 3 million steps it's got to go, right? Just for one cell. When that process finishes off, hopefully the right side looks identical to the left side, and both cells that get this instruction will have the same traits. Again, though, you can see probably that sometimes mistakes are going to be made during this copying process. Even though it's simple, mistakes can be made, and that's not always a bad thing. Sometimes mistakes are a good thing. They lead to variety and diversity, so that we're not all the same. And if we were all the same, what kills one would kill all, and that would be bad. So diversity and variety is good, and it comes from mistakes made from DNA replication. So kind of interesting. So at the end, 2 new DNA molecules form each containing 1 new chain and 1 original chain. Last thing for today is right here. This is it.
One of the most beautiful things about the structure of DNA is how it replicates. DNA replication is how the DNA makes an exact copy of itself, which it needs to do before the cell can divide. First, DNA helicase unzips the DNA by breaking the hydrogen bonds between the bases. Then, DNA polymerase adds complementary nucleotides to the correct bases in the original DNA strands and connects them. The complementary strands always match A with T, C with G. So if you know one strand, you can predict the other. If the original strand has these letters, the complementary strand will match T with A and C with G. It turns out that DNA replication happens smoothly and continuously on one strand, but in small broken sections in the opposite direction on the other strand. The continuous strand is called the leading strand, and the one built in sections is the lagging strand. The reason for this method of replication is that the bases must be added in only one orientation, sort of like the way Lego bricks have to be linked together in only one orientation. The lagging strand sections are connected with an enzyme called DNA ligase.
Okay, remember quickly, guys, that DNA is like a double lane highway, right? One or double lane road, one lane going, northbound, one lane going southbound. In your vocabulary assignment and in your textbook, you'll see something called Okazawa fragments, and those are these sections of DNA being added chunks at a time. Whereas on this strand, let's call that the northbound lane, traffic flows continuously. You don't have any breaks in the system. But if you again notice, we're going to go from having one double-stranded original molecule, one old DNA strand or double-stranded molecule, to having 2 double-stranded molecules, but each double-stranded molecule is going to be half old and half new.
And then the copy is complete. Errors in DNA are very rare, only about 1 in every billion base pairs. That's incredibly efficient, and it's a good thing that it is. Mutations can be harmful or even fatal. The enzyme that checks base pairs and repairs mistakes is DNA polymerase. It's the same enzyme that links DNA bases together during replication. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe and follow me on Twitter at SciencePet.
Okay, you heard her say that mutations can be bad. Sorry. You heard her say mutations can be bad, but which is true, but mutations can also be very, very, very good. So, all right, that's it for today, folks. Stay safe, stay healthy. I hope I will see you all soon. For now, signing off, Mr. Dunbrook from his basement, and stay tuned for part 3 of this PowerPoint, and that's going to include something called transcription and translation of DNA, which is really how DNA leads to your traits. So stay well, everybody. Sorry this video was so long. I understand if you had a hard time getting through it all at once. All right. Have a good day. Bye-bye.